Hi guys, my name is Gavin Emdor. Today we're going to be talking about a study called a GPR35 tuned gut micro brain metabolic axis regulates depressive-like behavior. I'm really excited to show you the study. I hope you guys enjoy. So, our bottom line up front here is that this study connects genetics, the microbiome, and brain function into one unified model of depression. It identifies specific bacteria and molecules that can be potential targets for antidepressant therapy, and it's one of the few studies when studying depression that translates findings from mice to humans. And this makes it very clinically relevant. Not infrequently are we able to apply our studies from mice directly to humans, but we actually do have a very interesting direct connection here that I can't wait to show you guys. So, just some background here. This study is all about expanding on what is known. So, it's like, for example, I think all of us know that, yes, the gut it can actually influence how we feel. We know the gut can influence depression. However, we don't know exactly how. The mechanisms for this connection are still being studied, and this journal article, this study proposes a mechanism that is shown to contribute to the development of depressive-like behavior. And just some abbreviations: NAC, nucleus accumbens; MDD, major depressive disorder; PD, we're going to be talking about this a little bit, parabacteroides dystesonis. So, what's the need for studies like this? Well, in 2020, an estimated 14.8 million US adults aged 18 or older had at least one major depressive episode with severe impairment in the past year. This is from ADAA.org. But we also know that bipolar disorder, autism spectrum disorder, and anxiety have all been found to have links to the gut microbiome. Let's just look at an example here from the study, the fecal microbiome and metabolome of Pitt-Hopkins syndrome, a severe autism spectrum disorder. So yeah, as the title says, PTHS, Pitt-Hopkins, is a severe form of autism. And they actually found in the study that individuals with PTHS hold a higher load of Clostridium boultiae than their parents, as well as people with both more typical autism spectrum disorder and people without autism spectrum disorder, showing that there is a microbiome component that comes with this disorder. They also found that specific metabolites were associated with Pitt-Hopkins, including bile acids and sphingosines. But there's also the fact that not many gut-brain studies, as I said before, are expanded into humans, and it's essential that we do this and continue to do this in order to be able to confidently say what the effects that our gut has on our mental state and our physical state. So a big part of this article is all about it strings many areas together. So it's all about GPR35. GPR35 is a gut G protein coupled receptor. So a G protein coupled receptor is I kind of like to consider them a type of, bouncer to a cell. When activated, they trigger these big pathways that have, for example, it can be an inflammatory pathway, or they have found to actually be anti-inflammatory. Its function is pretty diverse. But the GPR35 has been established in this article to be connected to changes in the microbiome that influence neuroplasticity. Neuroplasticity is kind of our brain's flexibility. It's like if there's like examples of people getting shot in the head and parts of their brain are damaged, yet they're still able to completely function. That's neuroplasticity. Our brain rearranges how it functions, but it can also go the other way. Plasticity can be disrupted. And we see this here through increased levels of Parabacteroides dystacionis. And so the study, it connects stress, it connects gut lining receptors, gut bacteria, microbial metabolites, gut plasticity, and from that mood. Alright, so now we're going to talk about the methodology. There is use of both mice and human subjects. Mice were used with and without the deleted GPR35 gene, and they were subjected to behavior tests. These consisted of open field tests, social interaction tests, the tail suspension test, and the forced swimming test. These tests were also performed, but we're going to be focusing on these. I gave you the abbreviations because they do come up a lot in the figures. So various iterations of these tests occurred as the experiment continued, as they played around with different parts of the pathway they were trying to prove. And then retrospective tests, primarily fecal microbiome sampling, were performed on the human subjects following the examination of mouse-derived data. And then all 16S rRNA samples were sequenced through Illumina. So now we're going to talk about our first set of results. So these figures can be a little confusing, so I just want to help. I have text kind of summarizing it, but you don't really need to focus on those. It's just listen to me and you should be okay. So basically what happened was that they had wild type and they had knockout mice. So basically the wild type, perfectly normal mice, nothing happening with them, really. But the knockout mice, well, they had the GPR35 gene, this receptor we're talking about, they had it knocked out. So that's why I'm going to use the term knockout. So what happened is that both of these mice were subjected to a series of stressors that have been shown in the past to create depressive-like symptoms in mice. So following this, what we see is pretty significant. Mice, the wild-type mice in an open cage roaming test, they're all over the place. You see this? So they're walking around. It's kind of like it tracks where they walked. So we can see that they're walking all around the cage. While the knockout mice, they are consistently staying on the sides, maybe going into the center a couple of times. We can see here, there's a significantly less amount of center time for the knockout mice. Significantly less center entries. There is more immobility time during tail suspension, and they also have a significantly lower social score. So we can see that when you take out the gene from a mouse, it increases depressive symptoms. Okay, so what the researchers then go on to study is what if they're just saying, what if we take out GPR35 from just the gut? So to do this, they do a little bit of genetics magic. This figure is a little more difficult to explain, but basically they crossed GPR35 flox mice, which they just have these receptors. In these mice have special receptors implanted into them through genetic modification, and they were crossed with Villain-Cree mice. So basically the Villain-Cree allows for any GPR35 but only in the gut to be deleted. It leaves the rest of the body unaffected, and this way we can see if there are any effects that can be tied directly to GPR35's role in the gut and not anywhere else in the body. It gives us a clear look just how does the gut influence this, and we see a lot of similar results. So in the gut knockout compared to in this case the wild type, we see much less, and we see significantly lower time in the center. We see much higher immobility time. The asterisks mean significance. This is definitely significant. We also see significantly lower social score. But on top of this, we also see that there's differences through Golgi staining. We see that there is reductions in the neural spine density of the nucleus accumbens when GPR35 is lost. So as you can see, it's like it's much thinner when there's a knockout, and you can see here, but it's not as profound. But basically, the nucleus accumbens is kind of it's part of our reward pathway, and what this shows us is that there is and it shows us that there is a change in our reward pathway when the GPR35 gut it's manipulated. We also see and this data is also just shown here, but we also see that dendritic branching is reduced in both GPR35 full body knockout and gut knockout. So there is absolutely a correlation of loss of the gut GPR35 to depressive symptoms. So we're building our chain here. So let's keep on going. So next up, this slide is all about connecting the communication of GPR35 to bacteria to the brain. We've done GPR35 to the brain. Now we're adding another piece in bacteria, the microbiome. So based on metabolite set enrichment analysis, or MSEA, we can see over here, sorry, let me get there we go. We can see here that, okay, so they looked at 107 differential metabolites through untargeted metabolomics, and they focused in on the top 4, and out of these top 4, they selected tryptophan metabolism. So basically, tryptophan is has a lot to do with our mental health because it's a precursor to serotonin. Serotonin is kind of like one of those feel-good chemicals. It has a lot of effect on us. There have been studies that have shown that lower amounts of serotonin can increase depression. And in fact, in a study called The Effects of Dietary Tryptophan on Affective Disorders, they show that increased consumption of tryptophan results in less depressive symptoms in patients with depression. So what they see here is that something called IALD, which is basically it's called indole 3-aldehyde. It's a metabolite of tryptophan. So when tryptophan is broken down by certain microbes, this gets created. But they actually noticed that it was significantly lower in the colon, the nucleus accumbens, and the hypothalamus of GPR35 deficient mice. So here we can see for the colon and this just shows that data. But how do we know that GPR35 is involved in this? Well, we don't know the exact mechanism, but we can see that in the wild type, compared to the GPR35 knockout, there is a significant difference in not the alpha diversity, which is what types of bacteria, but the composition of them. So there's increased levels of certain bacteria. Yeah, so let's go on to the next slide. So next up, the researchers did fecal microbiome transplantation. I think we know what that is. I don't have to get into it. But basically what they did was that what these researchers did is that they gave mice wild type just normal mice an antibiotic cocktail, and then they did a fecal microbiome transplant, and then they did behavior tests. This is kind of like the timeline here. So what they did is that some of the mice got a wild type microbiome, while some of the other mice were GPR35 knockout recipient. They got a microbiome from a GPR35 recipient who had been subjected to the stressors that causes the depressive symptoms. So as we can see here, previously normal mice, there were no stressors performed on them. They are showing just the same patterns of lower center time, lower social score, and spending just less time like crossing into the center more. So just by getting the bacteria into their gut, is enough to cause these depressive changes. So next up, the researchers did a shared environment experiment. So basically, what they did is that they had wild type and GPR35 knockout mice. They were co-housed together as littermates starting at 6 weeks old. The theory is that the mice would naturally swap gut microbes through grooming and just being in the same environment, and this would lead to microbial exchange over time. So after 8 weeks, they found that, as we can see here, this is just kind of a similarity display of the microbiomes of the wild type single housing and the GPR35 single housing. And then after they're co-housed together, look at this. Much more overlap. So the theory was correct that they would kind of merge together. So we actually see here, so let's just look at this. So wild type GPR35 knockout just housed separately. But then once wild type is with that's not significant. Once GPR knockout is alone, after it's with a wild type, its immobility time on tail suspension it's decreased. Immobility time for wild type single housing after it's co-housed, it increases. So we're seeing a sort of we're seeing in both directions, they both go separate. In both types of mice, they both go the opposite way. And even there's lower social scores as well. So we're seeing improvement in GPR35 knockout after being with wild type, and we're seeing debilitation of wild type mice after being with GPR35 knockout. So I know that's a lot, but hopefully you understand. So next comes the evaluation of the bacteria. So basically, they looked through the researchers, they looked through bacterial species that were consistently altered in both the donors and the recipients. And some of the top candidates included Parabacteroides distassonis, PD, Parasittarella excrementa hominis, I'll call that PE, and Clostridium scindens, let's go CS. So what they did is that they found that in CS that it actually increased it actually reversed depressive symptoms. They found that it was reduced in GPR35 knockout mice. They tested whether adding it back would improve mood. they did find this. They found that CS colonization reduced anxiety and social avoidance, showing antidepressant effects. So that's extremely interesting. Um, so what they did next is that they looked at PD and PE. So they used pasteurized or killed versions, along with just a pure control, um, along with live versions of both of these bacterias. So we can see there are in these saline compared to the pasteurized version, there really isn't any difference in any of these tests. However, when we compare both of these to live cultures of both PD and PE, there is significant differences, as we can see here. There's also differences in social scores as well, and yeah, but what they also found was that serum levels of IALD were significantly lower in PD colonized mice, and ILA, which is just indole 3 lactic acid, was found to be significantly decreased. So basically, IALD and ILA are kind of like two ends of the tryptophan metabolism chain. So we're seeing that there's sort of a preference, when there's higher levels of PD towards ILA. So, but then they found that increasing microbial IALD, that reverses behavioral changes of the knockout mice, while increasing ILA will increase the amount of depressive symptoms. So now we're adding another thing to the chain. So we have loss of GPR35 to increased PD to differences in metabolism causing depression, but we still have a little bit more to go to complete this chain. So next we're looking at neuroplasticity. This is an example of neuroplasticity in this picture. It's basically just showing that the same brain can really brains can look different, and that they can have very different functions based on the person. So what they did was that RNA sequencing was performed on brain regions suspected to be genetically regulated by tryptophan metabolites. And what they actually found was IALD was found to enhance synaptic plasticity and antidepressant gene expression, while ILA was found to suppress these same pathways, promoting depression-like changes. So these findings show us that we have a full chain now. We have loss of GPR35. We have increased levels of PD, disruption of metabolites, and then we have expression of depression. There is other studies that show how IALD is actually implicated in this. In the association, it's a study called The Association Between the Indole Pathway of Tryptophan Metabolism and Subclinical Depressive Symptoms in Obesity. They actually marked the association between markers of the indole pathway of tryptophan metabolism and how IALD, decreased amounts of it, is implicated in depressive symptoms in those with obesity. Yeah. So next, the researchers moved to human subjects. So this is where things get really interesting, in my opinion. In blood samples from 85 MDD patients and 77 healthy controls, they found that in the MDD patients, IALD was significantly lower in their bloodstreams. And then in this separate clinical data set, they studied the fecal microbiomes of these same patients. They found that PD was significantly more abundant in MDD patients. So this suggests that having increased amounts of PD can cause it could possibly be implicated in depression. So it's not just oh, you get stressed and then you get depressed. You get stressed, your gut microbiome is influenced, and then you get depressed. It's another part of a chain. But, PD has been observed to have positive effects as well. There's a paper that I'll be attaching at the end that suggests it can actually help prevent colon cancer. This just goes to show the microbiome is so complex. We can only help but to understand some of it. Hopefully I could help with that. And they show in that study that sorry, never mind. Okay, on to the next slide. Okay, so basically I'm just going to kind of sum everything up here. So if this shows that depressive patients a study shows us that depressive patients with higher levels of PD, this could be due to a GPR35 deficiency. GPR35 deficiency, we've shown, will increase levels of PD, causing different metabolism of tryptophan, which can disrupt this balance, as we see here, causing increased levels of ILA, causing debilitation of the neural spine of the nucleus accumbens, and then this leads to depression-like behavior. And so there's a couple more pieces to this that I can't explain just due to time, but I hope you guys can understand this, and it's just really interesting. All right. So this study shows us that one of the routes from stressful stimuli to MDD involves a microbiome-mediated mechanism, particularly involving the bacteria Parabacteroides disdasonis. It gives us a new perspective in how bacterial metabolites can influence neuroplasticity, and with that, pathogenesis of depression. It allows us to further understand the mechanisms of the gut-brain axis. However, this is one of many possible discoveries. There's still a ton more work to do. In conclusion, studies like these show us more about the mechanisms that cause depression through how the gut microbiome works. While it is complicated, it is significant that this study is able to connect a process studied in mice and gain more insight about the same thing in humans. However, this is only a piece of a giant puzzle in determining a complex disease, the cause of a complex disease such as depression. Here are some questions that we can consider. How could this knowledge be used to help treat depression? What role does PD play in other organisms? Can this knowledge be used to help treat other species that display depressive symptoms? In what other ways does our gut microbiome influence the gene expression in our brains? Should the microbiome be more substantially discussed when treating patients with mental disorders? All right, here's my work cited. Thank you so much, and yeah.